Okay, so the disorder I'm going to be talking about today is dopamine beta hydroxylase deficiency, or DBH deficiency. And this disorder is an autosomal recessive inherited disorder that's caused by specific mutations, usually missense and nonsense point mutations, within the gene that encodes for the DBH protein. And the wild-type version of this protein is primarily involved in the conversion of dopamine to norepinephrine, so the defective protein that results from these mutations is going to lead to the inhibition of this conversion, which is primarily going to cause dysfunction within the autonomic nervous system. Now, the wild-type form of dopamine beta hydroxylase is found in two general locations. The first is going to be in secretory vesicles of adrenergic neurons. DBH, as well as the molecule it creates, norepinephrine, is going to wait inside these vesicles until they're signaled to be released. And then when they're released, norepinephrine can then pass on these signals to adjacent neurons. Its second location is going to be in chromaffin granules of adrenal cells. And adrenal cells are located in the adrenal glands on top of the kidneys, as you can see on the left there. And DBH is specifically found in cells within the part of the adrenal glands called the adrenal medulla. And like in neurons, DBH and norepinephrine will wait inside these granules until they're signaled for exocytosis into the extracellular space. Okay, so how does the protein get there? Well, there's 5 general steps. Generally, a molecule called the signal recognition particle is going to target the immature protein to the rough endoplasmic reticulum, where it's going to be synthesized into the lumen. And after it's properly folded in the rough ER, the protein will be passed on to the Golgi, where it will be processed and sorted into these secretory vesicles, as stated before. And then these vesicles will then wait in the cytosol until they're released. So if we look at the first step in a little more detail here, we can see that in the cytosol, the first region of the DBH protein that's going to be translated is going to be the region called the signal sequence. So the signal sequence is going to consist of several positively charged amino acids followed by a hydrophobic alpha helix. And once this region is exposed or translated by the ribosome, the signal recognition particle is going to bind to the signal sequence and the ribosome, which will then induce translational arrest. And this will then target the SRP ribosome protein complex to the membrane of the rough endoplasmic reticulum. So once this protein complex arrives at the rough endoplasmic reticulum membrane, the signal recognition particle is going to bind to a receptor called the SRP receptor. And this will then bring the ribosome protein complex together with a protein that also is on the membrane called the translocon. And the translocon is actually going to act as this pore for the newly synthesized protein to travel through. So once the ribosome protein complex comes together with this pore, the ribosome is going to dock on top of the translocon. And this docking will then release the SRP and the SRP receptor. The positively charged amino acids that form the end of the protein signal sequence are then going to bind inside the translocon, and then this will initiate the proton to start to be synthesized into the lumen of the rough ER. And then once it's fully synthesized, an enzyme called signal peptidase will cut off the signal sequence, and then the newly formed protein will dissociate from the ribosome and float into the lumen of the rough ER. So once it's in the rough ER, DBH is going to undergo proper folding and many other post-translational modifications before being packaged into vesicles to be sent off to the Golgi apparatus. Now these vesicles are going to be COP2 coded vesicles, and COP2 is a protein, and that's going to help DBH exit the rough ER via exocytosis and then enter the Golgi via endocytosis. So while in the Golgi, DBH is going to go through many more post-translational modifications, such as glycosylation, before being packaged into these secretory granules that I keep talking about. These granules are going to have DBH and the norepinephrine it produces. As I said before, and these will reside in the cytosol until something signals their release from the cell. So in the case of dopamine beta hydroxylase, this is usually going to be an action potential that causes an influx of calcium into the cell, which will then signal the exocytosis of the vesicles. Okay, so as hinted at earlier, the primary signaling pathway that dopamine beta hydroxylase is involved in is the catecholamine synthesis pathway. And this pathway involves a conversion of L-tyrosine, which is an amino acid that we usually get from our diet, specifically proteins in our diet, to catecholamines such as norepinephrine and dopamine. As you can see in the mechanism below there, DBH is specifically involved in the conversion of dopamine to norepinephrine. Now, this pathway is important for a number of reasons. Norepinephrine and dopamine are neurotransmitters involved in signal transmission through neurons in the nervous system, especially within the autonomic nervous system, which will then help regulate various bodily processes. For example, norepinephrine specifically is involved in muscle contraction and blood sugar regulation. And then in adrenal cells, norepinephrine is released as a hormone to control our fight or flight response. So it's going to help regulate things like blood pressure, heart rate, and digestive functions. So this is the structure of the wild type form of dopamine beta hydroxylase. It's going to consist of 4 main domains: the dopamine beta monooxygenase N-terminal domain, or the DOMAN domain. It's going to have a catalytic core with 2 copper-containing domains, CUH and CUM. And then it's going to have a C-terminal dimerization domain. Now, the function of the DOMAN domain in DBH is still being discussed, but it has a leucine-rich binding pocket that's thought to be a binding site for several likely molecules, such as ascorbate, fumarate, dopamine, or norepinephrine. The CUH domain is going to be a beta sandwich formed from 2 antiparallel beta sheets, 4 and 5 strands. And then the CUM domain is going to have a similar structure. And those are thought to be involved in oxygen binding and electron passing. So it's thought that oxygen binds to the CUM site, and then CUH is going to receive electrons from ascorbate and pass them on to CUM. And then lastly, the dimerization domain is going to consist of 2 antiparallel alpha helices. So these are just some other important structural features of this protein. It has highly conserved histidine residues that primarily reside within the CUH domain. The H stands for histidine. And then it has asparagine residues that exist in the DOMAN and the dimerization domains. And then these are going to serve as sites for post-translational glycosylation, as mentioned earlier. And then there's that leucine-rich binding pocket. Okay, so this is the reaction that dopamine beta hydroxylase is specifically involved in. As the name suggests, it's involved in a hydroxylation reaction, which is a type of oxidation reduction reaction, where an OH group is going to be added to a molecule. So we can see here that copper will be reduced from Cu2 plus to Cu plus, and then the reaction will oxidize ascorbate to dehydroascorbate. And in the end, the hydroxyl group will be added to dopamine to make norepinephrine. Okay, so DBH gene is located on chromosome 9, and it's going to consist of 12 exons. Most of the mutations that are involved with DBH deficiency are going to be nonsense or missense mutations within these exons. This disorder will have an autosomal inheritance pattern, and it's also considered an incredibly rare disorder, and actually only 25 individuals have been reported in literature. And it's likely more prevalent than expected, but it's also worth noting that parents who have the mutant genes have shown a history of miscarriages, so it's also possible that the gene simply just might be dying out. Okay, so here are some of the most common mutations associated with DBH deficiency. Mutation number 1 is going to be the most commonly reported in literature, and that mutation is within a splice donor site, which will cause improper splicing and a premature stop codon. If we look at the third mutation here, alanine to glutamic acid, this one is going to cause improper protein folding. And there was this interesting study by Kim et al. That demonstrated the consequence of this mutation using immunofluorescence. So they grew 2 cell cultures, one with the mutation and one without the mutation. And then they tracked the location of the protein against the location of the rough ER and the location of the vesicles, which is where the protein is supposed to be. And if you look on the left here, they found that the wild-type protein co-localized with the vesicles, and you can see that in the merge in yellow, which meant that the protein was in the correct location. And then on the right, they found that the mutant protein co-localized with the endoplasmic reticulum, which is going to suggest that the protein was stuck there. So as we learned, proteins can't leave the rough ER until they folded properly. So researchers actually hypothesized that this was the reason for it being stuck there. So they introduced an artificial chaperone to cells, and they found that the chaperone was very much upregulated when the mutant DBH was expressed. So yeah, this was kind of the first study to find out the consequence of this specific mutation. So ultimately, all these mutations are going to lead to a deficiency in the DBH protein. And as a result, this is going to cause an accumulation of dopamine and then a deficiency of norepinephrine, which is going to be the primary cause of most of the apparent symptoms. So the 5 most common symptoms are going to be bilateral ptosis, which is drooping of the eyelids, rhinitis, which is inflammation of the nasal passages, hypoglycemia, which is low blood sugar, body temperature dysregulation, and then orthostatic hypotension will be the most common symptom, and that's going to be a drop in blood pressure when standing. So in the case of orthostatic hypotension, DBH is involved in vascular muscle cells, where norepinephrine is going to facilitate vasoconstriction. So ultimately, the DBH mutation is going to lead to low blood pressure, especially when standing, and then frequent fainting spells. And then itosis is going to be caused by a similar mechanism, but in this case, norepinephrine will act on smooth muscle cells on the eyelid, specifically the superior tarsal muscle. And as you can see on the right there, it's just going to cause the eyelid to droop over the eye, basically. But within the case of DBH deficiency, it'll cause bilateral ptosis, whereas in the case of this picture, it's unilateral ptosis. So most of the treatments for dopamine beta hydroxylase deficiency are going to involve replenishing norepinephrine because it's the cause of most of the disorder symptoms. The primary treatment is L-DOPS, or L-3034 dihydroxyphenylserine. And this molecule is a prodrug that can be converted to norepinephrine by dopa decarboxylase, which bypasses the DBH step entirely. And this drug seems to work well for alleviating symptoms, but it doesn't get rid of them entirely. So I think future treatments, should focus on replenishing the wild-type form of the protein itself rather than the downstream products. And here's my literature cited.